Before we uh, continue though, does anybody have questions from yesterday? We did sequencing yesterday. Alright, okay, cool. So today we're gonna talk about DNA fingerprinting. So, before we go into exactly what DNA fingerprinting is, we call it DNA fingerprinting, but what we're actually referring to is the fact that our fingerprints are unique to us, and there's a way to determine that your DNA is like a fingerprint. Your DNA doesn't have an actual fingerprint, but you can analyze it such that your DNA is unique to you. But how? So, there is a lot of variability in our DNA when we compare ourselves to another person. Is it true or false?
I think it's mostly the same.
Yeah, so that's true. It is mostly the same. Are there more non-coding regions than coding regions in our DNA? True or false?
I think true.
Yeah, so this is actually true. I don't know if you guys have learned this yet, but it's true that there are more non-coding regions than there are coding regions. And just to give an answer or just to give an explanation to the second question, it's like how we can say that we have what 90 plus percent DNA that we share with um what was it, other primates, or we share a lot of our DNA, or there's a lot of similarity in our DNA to, like, let's say other primates, or even like a large percent of our DNA can be compared to bananas. Have you ever heard that? Have you ever heard anybody say that? That's because for a lot of our DNA, the stuff that codes, or the genes that are coding, actually make up a pretty small percentage of the total DNA. So, next question. The DNA regions that are the most different between individuals are the coding regions. True or false? Any guesses?
True, maybe? Or just
Uh,
like what is being coded for? Like, I don't know.
so here, I think a lot of these questions are trying to trick you. So, coding regions, let's say, are your genes. So, I have two hands and 10 fingers. How many of us have two hands and 10 fingers?
All
Like,
of us.
like probably a lot of us, right? Most of
Majority
us.
of population.
The overwhelming majority. How many of us have two eyes, two ears, one nose, one mouth, two legs, two arms? So actually, coding regions between people are quite similar. So we can say things like hair color or, eye color, skin color. Like these can be differences in coding regions, but overall, it's a very, very small difference. The most different regions are actually the non-coding regions. So the non-coding regions see the most variability between people. So DNA fingerprinting, last question, looks at our genetic sequence to differentiate between individuals. True or false? This one should be pretty easy.
True.
Yeah, true. It doesn't look at our actual fingerprints, right? It looks at our DNA. It looks at our genetic sequence. So, what is DNA fingerprinting? So, it is a technique developed by Alec Jeffreys in 1984 in Leicester, England. It is a method that identifies an individual based on the patterns
Mic
formed
feedback on.
from the variations in the genetic code. So it's also known as DNA profiling, genetic fingerprinting, or DNA typing. So this is a little video about the first criminal case ever solved with DNA fingerprinting. Um, you guys can watch this on your own time though. So what happened? A man named Colin Pitchfork was arrested and then convicted on the rape and murder of two teenage girls in 2 different years based on DNA fingerprinting. So this was the first use of DNA fingerprinting to convict a criminal. Yeah, and it was the first. He was the first man to be caught as a result of mass DNA screening. So, a bunch of local men volunteered their DNA, and then there is like an interesting situation between like some man being paid by Pitchfork to masquerade as himself, and there's another man whose innocence was established with DNA fingerprinting in the same case. So let's look at the next slide. All right, so this is a little pie chart of the human genome. So if the entire pie chart is all of our DNA, if you look here, the protein coding genes make up 2% of our entire genome. We have 20,000 to 30,000 genes, but most of the DNA does not encode for a protein or RNA. It's not a gene. So look here, 26% introns. Um, Lines are long, interspersed like oh, I forget the name. We're gonna go over the names actually, I think, in the next here we go. So introns, and then we have regulatory sequences, and then we have highly repetitive sequences, um, interspersed repetitive DNA, transposons. Okay, yeah, okay. Give me a second to look at this pie chart again. All right, yeah, all right. So the main thing to take away from these slides is that of our entire genome, a very small amount of it is coding. So when we want to look at somebody and create a DNA profile from them, the main the main points that we look at won't usually be with these protein coding genes. So these protein coding genes won't really be different between people. What we have to look at is the rest of these different types of sequences, and these are where you will find variability between people. So if you look here, this is a little pie chart of the different types of non-coding DNA. So everything that's not this 2%, basically. So you have introns and regulatory sequences. So introns, and I don't think the regulatory sequences have a separate section here. And then you have something called highly repetitive DNA. So yesterday, if we look, remember when we looked at the shotgun sequencing and the problem was with the highly repetitive DNA, there's many different types of highly repetitive DNA. We have telomeres, which we have already learned of, tandemly repetitive DNA. So what that means is a certain sequence is going to just keep being repeated a certain number of times in tandem. And then something called interspersed repetitive DNA, which is just interspersed just means like spread, like sprinkled at different points on your genome. It's interspersed and it is repetitive. And another example of this is transposons. So today we're going to be talking about tandemly repetitive DNA because these are basically the sequences that you guys would have looked at if you went to the Ontario Science Center and done the fingerprinting there, the fingerprinting activity. So these guys are also known as VNTRs, variable number tandem repeats. So these are short, identical DNA sequences that are repeated in series. An example would have been the D1S80 locus, which you guys would have looked at the Science Center. The repeat length of this sequence, or this repeat, is 16 base pairs. There are many types of VNTRs, and they're most often classified by the length of their repeat. So a short tandem repeat is a subcategory of these VNTRs where the repeat length is very short, or short, most commonly 4 base pairs long. And another example would be the D10 S1248 or S1248. So what are these VNTRs? Majority of them are non-coding, so the differences between people are harmless. But there are some examples of coding repetitive DNA, like Fragile X or Huntington's disease, where the number of repeats determines your phenotype. What is Fragile X syndrome? It's the first triplet repeat identified, and the repeat sequence is CGG on the X chromosome, and the normal Fragile X allele is 30 repeats in the 5' untranslated region of the first exon. Okay. And Fragile X syndrome happens when you have over 100 repeats of the CGG triplet. And this is the most common single known cause of intellectual disability and autism. Huntington's. I'm sure you guys have already heard of this before. Huntington's repeat sequence is CAG. Oh, and what is it? It's a neurodegenerative disorder that affects muscle coordination and leads to cognitive decline and psychiatric problems. So, normally the individuals that are affected with Huntington's disease, they normally or typically have at least 36 repeats or greater. So if you look at this chart, it's basically showing you most of the time the number of repeats versus whether or not they're affected by the disease. Cool. So let's look at some other characteristics. They're polymorphic, so they have many forms, which means that they can have many alleles. So what does that mean? To put it into more simpler terms, when we looked at Mendelian genetics, you basically saw that you could have a dominant or recessive trait. So you could have like dominant or recessive, which we use little letter for. But when you have a polymorphic gene, you could have more than two alleles. So you could have like, um, let's say you have a 16 base pair repeat, you can have one repeat. You could have two repeats. You could have 10 repeats, or you could have 13 repeats. So if you look at Huntington's, you could have less than 27 or you could have up to greater than 39. So these are all different alleles, 39, 40, 41, all the way up to, on like 50 or 27. So this is what it means by polymorphic. What is this cause of polymorphism? Well, the allelic variation, like I said earlier, is due to the number of repeats that you can see. So the number of times the sequence is repeated is the allele, and this repeat sequence can be repeated many different number of times in the population. So this is just a picture representation of polymorphism. So if one box is one repeat, each little line here would be the chromosome at that location of a different person in the population. So if these were all the alleles that can exist, how many different alleles of this loci are present in this population? It looks like, 1, 2, 3, 4, 5, 6, 7, 8, 9. How many alleles can exist on one individual in this population? It looks like you can only have one allele because we're only looking at this location, and it looks like if you have 14 repeats, you have 14 repeats. You can't have 14 and then another like 15 or 16 following. Do you guys have any questions about this slide? Or the questions that it's asking here? How does the first question differ from the second question? The first question differs from the second question because,
Sorry, could you repeat the answer to the questions again?
yeah, so the answer to the first question, how many different alleles of this loci are present in this population? So what it means by this population would, I guess, just be all the people on this slide because each little strand here was taken from a different person. So to answer the first question, you just count the, like every person here seems to have a different allele, so you just count everybody, which is 9 people. So it looks like there are 9 different alleles of this loci present in this population. And the second question, how many alleles can exist in one individual? So what this is asking is how many variations of repeats can you have in one individual? So the answer that I would say is each individual can have one allele. And why is that? Because in each individual, we can see that if they have 14 repeats, then they have 14 repeats. If they could have 2 alleles, then that means that they could have 14 repeats and 15 repeats. But if you can just look at their DNA, they don't have a 14 repeat and then another 15 repeat following after. So in each loci, every person has one allele or one number of repeats.
Okay, thank you.
All right. Is there any confusion about the terminology I'm using? Does everybody understand what a loci is? And everybody knows what an allele is? All right. Okay, yeah, I can review. So a loci is just a specific location on the genome. So the way that they refer to it here is just like, okay, here, D1S80. So this is like DNA chromosome 1, site 80. So when they talk about loci, it's just a specific location on the genome. And they refer to it by DNA chromosome number and then the site number. And the site number is just, I guess, the site that they found first. If it's site 1, then it's the first site that they found. If it's like 80, then it's the 80th that they found. And this terminology is used to describe VNTRs. Yeah, they don't need to code for something to be called alleles. So VNTRs can have alleles because the number of repeats of a sequence that you can have is variable and it can change between people, or it's different between people. I guess an allele would just be a variation, if maybe that's not the best way to put it. An allele would be, let's say in terms of this D1S80 locus, a single repeat length is 16 base pairs. An allele would be if you have one repeat at this location, if you have three repeats at this location, if you have four repeats. And that would be similar to having, let's say you have brown eyes, or you have, I mean, that would be a phenotype. So I guess like dominant, homozygous dominant. No, that would not really be the thing, because then like a dominant allele, recessive allele. But here it's not by dominance, it's by the number of repeats you have. That's how we say what an allele is for these tandemly repetitive DNAs.
But if you scroll up, it says that if DNA doesn't code for something, then it's not called a gene.
Exactly.
But it can be called an allele.
So we don't call these VNTRs genes. What determines whether or not something is a gene is whether or not a protein comes from it, or RNA.
Okay.
If DNA encodes for a protein or RNA, so if you will transcribe that region of DNA, it's considered a gene. Alleles are essentially, let's say, give me a second to think about this. The best way to describe what an allele is. Here, maybe I can Google it and then give you the best way. What does the internet say? It has gene in it.
Oh no.
No, like it doesn't have to be a gene because for our purposes, we are saying that there are many alleles because for these VNTRs, between people, you can have a different number of repeats. So it's essentially like your allele is one repeat at this specific location. Another person's allele could be, they could have, 10 repeats. But let's talk about that in terms of a gene that you might think about. So your eye colors. So you could have alleles of dominant, recessive, or homozygous dominant, homozygous recessive, right? So all the times before, we've talked about alleles in terms of dominant and recessive alleles for Mendelian genetics. But in this case, it's not about dominance or dominant and recessive. It's about the number of repeats you have. And your allele is basically how many repeats you have at that location. It's just a characteristic, I guess. It's just the characteristic that you have.
All right.
You don't have to. It doesn't have to be a gene for you to have an allele at that place. Because if there's
All
variation
right.
between people, then these two people have different alleles, right? Whether or not it's encoding for a protein or RNA. And that's the point. That's just the main takeaway here. All right?
Okay.
All right, cool. Let's move on. Okay, so let's look here. Let's look at these examples of polymorphism. D10S1248, D1S80. The characteristics of these two loci. What is their repetitive sequence? The length of the repeat, and the length of the repeat can be found by multiplying the number of repeats times the length of the sequence. So this will be different in every person. The class of repeat. So remember we talked about STRs, oh, which is short tandem repeats. It can be long tandem repeats. The number of variations. So the number of variations can talk about, you have 8, 9 repeats, 10 repeats, 11 repeats. The number of variations is a different number. Basically, if everybody had a different allele, or if everybody had to have a different allele, then how many people can you have, essentially? So if you can have up to 8 to 19 repeats, and each person can only have one like number of repeats. So if you have 8 repeats, then you have 8 repeats. If another person has 9 repeats and they have 9 repeats, then yeah, 8 to 19. 19 minus 8 plus 1. Uh, that's the number of variations you can have. Cool. And PCR fragment length, why do we talk about that? We talk about that because we are going to use PCR to build your DNA fingerprint. Alright, cool. In a population, a polymorphic loci has many choices, or alleles. And in an individual, there are only a maximum of 2 alleles, because on each DNA strand, if you have, let's say you got 8 repeats from your mom and 9 repeats from your dad. Oh, so then that kind of changes what we learned about earlier, but yeah, so I'm guessing the number of variations isn't about people, it's about just on this one DNA strand. How many variations can you have? So, when you talk about an individual who has 2 DNA strands, it gets a lot more complicated. And this uniqueness, though, is important. And this uniqueness is what allows these VNTRs or STRs to be used to build a DNA fingerprint. So, which biotechnology could you use to most easily identify the differences between individuals here? What is the main difference? The length of this location, or essentially the length of the sequences at this loci is the difference. And length. Remember what can identify the differences in length between DNA fragments? Exactly. Jelly electro, Nathan says. Yes. Jelly electro. Gel electrophoresis. So, how do you use gel electrophoresis here? We are going to use PCR and gel electrophoresis to build your DNA fingerprint. Okay, so what do you have to do? First, you have to figure out which fragment of interest you are going to amplify. So, step 1, fragment of interest in STR, in this case, is at a specific loci on the chromosome. So you need to figure out which primers in PCR will define or locate the fragment of interest, and once you can do that, then PCR can amplify that fragment for you. And then afterwards, you can use gel electrophoresis to separate the PCR products by size and then visualize those DNA fragments. And the way this will work is, let's just draw it out. Let's say because you're gonna get a set from your mom and your dad, so I guess if you have like, let's say you have 8 repeats, and then you also have 10 repeats, then PCR, because you define the primers that will locate the fragment of interest. These segments, they end, they begin and end the same between people. It's just that the number of repeats is going to be different, right? So your primers can help you, and then afterwards you'll have many different fragments. But let's say you have like an 8 repeat length and a 10 repeat length, and then once you visualize on your gel electrophoresis, that will show up as 2 different bands. So what would you have done at the science center? Everybody would have taken their own DNA from a cheek swab. So this is, let's say, this is person 1, this is person 2, this is person 3. Person 1 looks like this on the gel electrophoresis. Person 2 looks like this. And then after that, how would you say person 3 looks? Any ideas? Actually, before we do that, why don't we figure out which direction the DNA fragments are moving? Is DNA moving down?
I think it's moving down, yeah.
And
Because
how do you know?
A's fragments are shorter, so they're closer to the bottom.
Exactly, so that's what you should be looking at. Good, DNA is moving down. And A and C, remember you guys learned homozygous or heterozygous? Is A heterozygous or homozygous?
Hetero, I think, because
Exactly,
that's two different alleles.
exactly. So they have an allele that is shorter, or one allele that's longer. I'm not going to count it out. Seems like too much work. But A is heterozygous. C is also heterozygous. Is B hetero or homozygous?
Homozygous, because they're the same size.
Homozygous. So when you look at their bands, are they going to be 2 bands, or is there going to be 1 band?
One band.
Exactly, one thicker band. And do you know where it might be? B's band?
Between the two C's?
Yeah.
Between the two C lines.
And how do you know that?
Just comparing the strand length.
Exactly. So you're gonna have like a thicker band. Yep. So each person will have 2 copies, but the copies can be different lengths or the same length. And but by copies, it essentially means, I think, allele. So if the copies are different lengths, then they're heterozygous, and if they're the same length, then they're homozygous. So, loci. If all the loci present are homozygous, how many loci are represented in this uh, like gel electrophoresis? Any ideas?
6.
Yeah, you're right. 6. And if all the loci present are heterozygous, how many loci are represented?
3.
3. Does everybody understand how we answer these questions? So the way that we know is, let's say they're heterozygous, then that means for every loci, you're going to have 2 different bands. So it's this situation. At this one loci, you have 2 different alleles, so you're heterozygous. So you're expecting 2 bands. So what do? We count.
Sorry, isn't a loci like a position on the DNA? But we're taking all these alleles from the same position, so how does that work?
So
Are they the same?
we are going to say that we have taken multiple positions.
Okay.
We don't know how many. We only know if the loci are homozygous or heterozygous. So it's the other way around of thinking.
But are we looking at the same allele now, or different alleles? Like different genes? Or I don't know what you call them.
So different alleles.
Different regions of DNA? Is this still the same region?
No, these would be different regions of DNA. Okay, so in this question, or in this example, you know if they're homo or heterozygous, and then you can say the number of bands that they have. This is just looking at it differently. This doesn't have to be at one loci, right? This is you don't know which loci this is. You don't know how many loci this is. You just know if it's homozygous or heterozygous. So this is just completely hypothetical. I guess. This is just a completely hypothetical question that's just asking you. At most, you can have two bands per loci. Does that make sense, guys? So my previous explanation, I think that's why you might be a little bit confused. I made a mistake. If you have like 14 repeats, that's it. You can't have like 14 and 15 later. The only way you can have 14 and 15 is if you have 14 on one DNA strand and then you have 15 on another. Does that make sense?
Yeah.
So at this loci, you have two options, right? You can either have both that are 14. Oh my god, that's so bad. You can either have both that are 14, right, and then the same length, or you can have two that are different lengths. If they're the same length, then you're homozygous, and if they're different lengths, then you're heterozygous. But what it's asking here is you don't know if this is one loci. You don't know if this is 3 or 6 or however many loci this is. You only know if you're homozygous or heterozygous. And in real life, you wouldn't really be able to have a question like this. It would usually be the other way around. But this is just hypothetically asking so that you understand that at each loci, you can have up to two alleles. But if you're all homozygous, or if you're all heterozygous, then what happens to the number of loci? Does that make sense? It's just a knowledge question. It's not really too based in reality.
Okay. Okay.
So if they're all heterozygous, like how would you know looking at a band that they're all heterozygous? You wouldn't really be able to know, right? But let's just say hypothetically they were all heterozygous. Then it would be from three loci. Because in real life you could have any number of combinations, right? Like this could be a homozygous band for a single loci, you know? These could be, let me erase, right? Like this could be a homozygous band from one loci plus, you know, different links, and this is another loci. Or you could even be this is a loci, right?
Yeah. Yeah.
So in real life you wouldn't be able to know, but this is just saying, you know, what if.
Okay.
So if they were all homozygous, then these are all loci. So then you have six. But if they're all heterozygous and in any combination, because you just can't have an overlap, right? So
Yeah.
any
Okay.
combination and then that's three. One, two, three. Cool. So allelic variation. The number of alleles at a polymorphic loci is pretty small compared to the total world population, right? So what does that mean? Like, there's a pretty good chance, like, the chance is not that small that, let's say I have 14 repeats. Like this person, the chance is not that small that in this call, another person at the same loci will also have 14 repeats, right? Because, let's go even further up. If you can have 12 variations at this loci, or let's say 28, which makes the number a little bit bigger, then in this call of 10 people, the chance is not that small, right? Or the chance is pretty large that one of us is going to have the same variation. But I guess since you have two DNA strands, the chances become smaller. But still, the chances are not that small that you could have the same alleles as another person. So how do we build a fingerprint, right? Because if an allele can be shared by 5 to 20% of all individuals, what implications would this have? And the implication here is that people can have the same alleles. So the true power of short tandem repeat analysis comes from looking at multiple short tandem repeat loci simultaneously. So if the chances of an allele being shared by individuals is 5 to 20% for a single allele or at a single loci, then if you look at multiple polymorphic loci, then the chances repeat or the chances of you having the same at multiple loci, well, that's really, really small, right? So in North America, 13 loci are amplified plus 1 to determine your sex. And if the chances of you having the same 13, the same alleles at 13 loci plus having the same sex as them. What about twins and stuff? Would they have the same alleles? Yes, identical twins have the same DNA. Yeah. So yeah, Nathan, you're right. Identical twins, well, even the chance of I'm pretty sure if you were wanted for a crime and you had an identical twin and you provided this identical twin's DNA, they might know that you have an identical twin. But yeah, chances are, unless you're identical twins with somebody, the chances of all your loci having the same alleles is really, really, really small, right? It's almost impossible. But it's not impossible. So if you ever do a DNA test, like for let's say it's a paternity test, and it's always going to tell you like 99.99% certainty. So the chance is really, really, really small, but I guess it's still possible. Okay, so polymorphism in variable number tandem repeats is useful for DNA fingerprinting and studying evolution. Both of them require looking at differences. So DNA fingerprinting would be the differences between individuals, but evolution would be the differences between species. So why do we use non-coding regions? Coding regions are too similar between individuals and even species. Because let's look at our primate, or yeah, let's look at our primate, other primates. They still have two hands, right? They still have 5 fingers per hand. They have 2 eyes, they have one nose, they have one mouth, right? So these coding regions are still quite similar between even species, right? But that's why we look at these non-coding regions, because it's in these non-coding regions that we really see variability. So let's look at some applications. I've brought up a couple applications already. Paternity testing, criminal cases, identifying a corpse, immigration disputes. Oh, I'm not sure how to use them for those. I have never looked that up. And food testing. Let's say, are you trying to determine if this is champagne or if this is horse meat? You can use DNA fingerprinting for this application. Let's look at paternity testing. Paternity testing is used to identify a child's parents, and that's because we know that each child inherits one set of chromosomes from each parent. So, let's look at here. Can you guys see the two like gel electrophoresis, the two like bands here in enough resolution to like tell them apart?
I think so. I think
Okay.
it's pretty clear.
Yeah.
So on the left you have paternity exclusion. So the alleged father cannot be the father of the child. And on the right you have paternity inclusion. So, can you guys tell the difference between the two of them? Do you guys need a little hint? Anybody know? The hint is the sentence on the previous slide. So if the
Wait,
child,
so,
oh yeah, go ahead.
for the alleged father, like the two bands don't match up with the bands of the child, but then with like paternity inclusion, one of the bands matches up with the child.
Okay. So, the hint was, yeah, you got it right. Each child will inherit one set of chromosomes from each parent. So what that means is when you look at a loci and you have the mother and the father, the child will, let's say, okay, well, I think we're looking, I, let's say that for all purpose, like for this purpose, we are looking at a specific loci where the people in question here, if they have two bands, they're heterozygous, and if they have one band, they're homozygous, but this is one loci, right? So that means that the child, if they're heterozygous, or even if they're homozygous, they will inherit one allele from the mom, and they'll have to inherit one allele from the father. In this case, paternity exclusion, it's not happening. In paternity inclusion, the child inherits one allele from the mother and one allele from this alleged father. So that's how they know that, well, okay, so this child could have inherited this allele from the father. So if you look at multiple loci, so the more loci you look at, better idea you can have, because it's still possible that if you only look at one loci, that this just happened by chance. But let's say you look at multiple loci and this is happening at every loci, then you can build a better idea whether or not the alleged father is the father of the child. So this chart shows 5 loci and the size of the fragments, so the number of repeats, through DNA fingerprinting of each individual. So just looking at the number of repeats of the child, the mother and the father, is it likely that the alleged is the father of the child?
I think so.
Yeah.
Okay, so how would you guys know?
In all of the second allele lengths, there's always an allele shared with the father on the right side.
Yep. So here, well, I guess sharing it with the mother is, already gonna happen. Like, you assume that. So the alleged father will only highlight it from here. 16, 16, it's present, and then 14, 14 present, 7, 7, 9, 9, 14, 14, and then 15, 15. So yeah, at every single loci, the child has inherited an allele from the mother and an allele from the father. So this alleged father, it looks like there's a pretty good chance that they might be the father of the child. So, I mean, this is just another example. If lane 2 represents the biological mother, so this is mom, determine which lane represents the father. Five?
Wait,
So,
which was the child?
so
Oh,
you
3. Oh,
have
okay.
to
Yeah,
determine which one
yeah,
is
5.
the child. So Nathan is
Yeah,
saying
3
5
is the
is the
child.
father. So how do you know that 3 is a child?
Because one
I
of
think
them is
either
the same as the mom.
I think 3 or 4
Oh,
can be the
wait.
child.
Oh, yeah,
It
or
could also be 4.
1 or 4. Yeah, it could be 4
Mm.
too.
So it can
It
be.
can't be. Oh, yeah.
One to 3 or 4 could all be the child.
Yeah.
Right? So Nathan says 5 is the father, and he said that pretty quickly. But yeah, is 3 the child if 5 is the father?
Yeah.
Okay, but let's say we didn't know the father.
Put that F somewhere else.
Alright, let's say we didn't know the father. Alright, could 4 be the child? Let's say we didn't know the father. Could 4 be a child to a different father?
I guess it could.
Yeah, it could, right? But I mean, you also have to look at the fact that 1 and 4 are the same, so, right? So if
But
we,
that could
whatever
just
we
mean
say
that
to
they're
1,
twins.
it applies to 4. So now let's look at this. How do we know that 5 is the father?
It has completely different DNA than the mom.
They
Nothing
could share
matches.
DNA with the mom still. So it's not the full marks answer that I'm looking for.
It has the same as all the kids that you have.
So for the child in question, because 1 and 4 both share DNA with the mom, so 1 and 4 can.
Couldn't one of them be the father and one of them be the child still?
Pardon?
Couldn't, like, if one was the father, couldn't 4 still be a child?
1 be the father.
If you
Okay,
weren't looking at 3 or 5.
okay. It looks like we have 3 children on our hands.
But we're assuming this is a family.
Exactly. I didn't assume this
Okay.
was a family earlier. So I asked the question weirdly. Okay, so there's 1 mother, 1 father, and 3 children. So then, okay, I see. So, how do we know that 5 is the father? And that's because for all of these children, you're gonna look for one band that comes from the mom, at least. So in child 3, this band has come from the mom, and this band has come from the dad. Child 1 and 4, they share this with the mom, and they share their other band with the dad. So 5 should be the father. And you can rule out the other options just by comparing them. So let's say that 1, we thought that 1 was the father. So what's the problem with thinking that 1 is the father?
For 3, there's nothing in common.
Exactly. So 3 couldn't be the child of 1. 4, 3 could also not be the child of 4. So that's basically how you would rule it out. Okay, this is just another question. I think we might have had enough practice. If you guys want to look over this, you are welcome to. Okay. Okay, let's look at this criminal case. You have collected a bloodstain from the site of the crime, and you have also collected the DNA from 4 different suspects. Which person is more likely to be suspected in the crime? David
John.
says John.
John.
Okay, you'll say John. So, how do you know it's John?
All of the bands are the same.
Yeah, so John is matching every band on the bloodstain. And just here, what does this DNA fingerprint tell you? If this is the blood from the defendant's clothes, this is the victim's blood. Is the defendant's blood present on the defendant's clothes? That's basically what you have to figure out.
I don't think so. There's like one band in the middle that's not there.
Oh, oops. So this band is missing from the defendant's jeans and shirt. This band is also missing. This band is also missing. So what can we say? We can say that the defendant's blood is not present on the victim's, or sorry, the victim's blood is present on the defendant's clothes, but the defendant's blood is not present on the defendant's clothes. But that's not what we're trying to determine here. Is the victim's blood present on the defendant's clothes?
Yes.
Yes, it
Yeah.
is. And how do you know?
All the bands are the same.
Yeah, all the bands are the same. So yeah. So what does it tell you?
The victim's blood was on the defendant's clothes.
Yeah, and that looks pretty bad, right? That would look pretty bad. How do you explain that? Okay, so this is a sexual assault case. They use DNA profiling for this too. This is a lot of questions, and I think you guys can probably go through this one on your own. That's the end of the lesson. Do you guys have... Oh, I'm not gonna scroll through that. Do you guys... Oh, yep, go ahead, Nathan. What was the control used for? Oh, okay. Let's see. Sizing marker, K562 control. I can tell you what the sizing marker is used for, but I wonder... that's a good question. Give me a second. Oh, what's the sizing marker used for? So essentially when we run these, like, when we run a gel electrophoresis, if... Oh, maybe I can go to the previous lesson and show you.
I think it just marks every possible interval that you can have
Exactly.
DNA end up at.
Yeah, so for the sizing marker, you know what you've put in here, and you know the base pair length of all these bands. So this lane exists here to compare to the lengths of all the other lanes to know the approximate base pair length of the other lanes. Yeah. And this K562 control, I'm not too sure why this is here. But I don't think it will affect your ability to answer the questions. They look like they're all the same too. Looks like they have the same alleles. So they must be from. Yeah, I don't know why they're here. That's a good question. But yeah, I don't think, again, I don't think it'll affect your ability to answer the questions here. Oh, it asks you here, what do you think the control was used for? Hmm. Give me a second. Maybe we should answer this together. I have no clue what the control was used for, and I've learned through
And
this already once.
it appears in 3 columns for some reason.
Yeah, they use it in 3 columns. So maybe we should ask Miss Law. I don't know what the control was used for. K562 cells. They are an erythroleukemia cell line. Oh, do one of the suspects or victims have leukemia? I don't know. That's a good question. Ask Miss Law. She might have an answer for you. I don't know why they use the control. I can tell you why they use the sizing marker, but I can't tell you why they use the K562 control. Any other questions? Alright guys, yeah. Good luck with this. If you have any questions about the fingerprinting, don't hesitate to ask here. If you have a question about the case here, don't hesitate to ask. Yeah, good luck studying and see you guys tomorrow. No, wait.
Thanks,
See you
Alex.
guys on Monday because tomorrow's Saturday.
Oh.
Losing track of the days.